Namaskar students, welcome to our channel Bio by Gulab Sir. As a part of series of lectures, today's topic is Animal Kingdom. As we already learned about Plant Kingdom. In series of lectures, it will be in the parts as Animal Kingdom is very vast topic. Today we shall just learn about classification of all the animals altogether, so that we will have easy to understand all the phylums, their characteristics and organisms belong to them. So let us begin with the classification chart, how whole animal world is being classified. This is the chart what I have been prepared here, that you can understand that whole animal kingdom is divided into two major groups, unicellular animal and multicellular animal. This is very easy to understand. But what is the term is being used for unicellular organism is protozoa, that is unicellular organism, and for multicellular organism metazoa. So please remember, in NEET question can be also an application of knowledge. So you must know that multicellular group of multicellular organism is known as metazoa, and unicellular as such you know what is protozoa, as you are familiar with Paramecium, amoeba, euglena, as they are protozoans. They are unicellular organism. They are easy to understand. But under the heading of metazoa, there are many groups or phylums that what has to be understood in detail. As the definition of metazoa is multicellular organism under which all the multicellular organisms are classified. But again they are divided into two major groups, that is what parazoa. This is the word is very important to understand by you. Parazoa and eumetazoa. This parazoa means multicellular with cellular organization, not tissue level organization. Parazoa means multicellular organism without tissue level organization or cellular level organization. And eumetazoa, that is what all the multicellular organism with tissue level organization. See, this is the evolutionary trend. Unicellular, multicellular without tissue level, and multicellular with tissue level. These are the stepwise. And yes, all the organism they have evolutionary history, and thereby that classification system is also in a sequence of their origin on the earth, and thereby you will enjoy this video when we will have detailed study phylum wise how that phylum is based or descendant for the next group of organism, and that what we learn. We will learn this characteristic as a evolutionary trend. That is the new way of learning to have a better understanding and best touch with the syllabus. So here you can understand parazoa and eumetazoa comes under metazoa. Now eumetazoa, they are tissue level organization. But next step to the tissue level is organ system and whole organ system level organization. Now please see, eumetazoa is further classified into all these 10 subphylums. Here one is coelenterate, second is ctenophores, platyhelminthes, ascahelminthes, so annelids, arthropod, mollusks, echinodermata, hemichordata and chordata. For detailed characteristic of each phylum, no doubt next video we are going to prepare, but before that, how they are being classified, that one step you understood, that multicellular with tissue level. Now here we have to have how these 10 phylums are classified under eumetazoa, on which basis they are separated. That is very important to understand. So let us see these ctenophores. They are multicellular with tissue level organization, but very important is that when they are multicellular with tissue level organization, why they are then separated from ctenophores? That is important. Why ctenophores are separated from coelenterate? That is important. So simultaneously we shall study that similarities and dissimilarities between each phylum, how they connected with each other in a evolutionary history. That is very interesting. Now very important, very base of classification, that's supposed to be understood, for which I am taking next page. Let you see what is that basic characteristic which is supposed to be considered for the classification is the presence of notochord and absence of notochord. So who are that non-chordate and who are that chordate, for which once again come to the first page. All except this chordate written here, these are non-chordate, including this parazoa also. Poriferas, coelenterata to hemichordata, they are non-chordate because they don't have notochord. See here, this is embryo structure. You will not find this type of structure what I have drawn here in this 3 diagram. Here, this is the notochord which present in embryo stage, in chordate, in non-chordate, notochord is absent. Hence, porifera to hemichordata, they are non-chordate. This is what one character that you understood. These group of phylums or animals belong to these phylums up to hemichordata. They don't have notochord in their embryo stage. Is it clear to you? So now second topic, When first is clear that till hemichordata they are non-chordate, and from chordate onward they have notochordate embryo stage, therefore they are chordate. Now among non-chordate, from cylindric to hemichordate, once again we have to understand why they are separated. For this, let you understand third diagram, what I have drawn here. Let you observe here. First of all, let you understand how this diploblastic coelomate, triploblastic acoelomate, triploblastic pseudocoelomate and triploblastic coelomate, how they are originated, how they are formed, what are they. Then you will understand who are diploblastic acoelomate, who are triploblastic acoelomate, triploblastic pseudocoelomate and triploblastic coelomate. You have to understand. Then only you will enjoy that. Otherwise, simply saying that porifera and cylindrate or cylindrate ctenophores, they are diploblastic acoelomate, okay, that word you will mug up. But unless and until you understood the meaning of what is diploblastic, what is coelomate, acoelomate, pseudocoelomate, you will not have idea. So let us learn first. These characters which supposed to be used for all the phyla, therefore it has to be understood. Now, as a part of evolutionary history also, this will be very interesting for you. You know that every organism's life begin from zygote. Is it? Am I right or wrong? Yes. You would be say yes, it is right. We all organisms, animals, their life is begun with the zygote if they are sexually reproducing. If they are sexually reproducing, fragmentation, regeneration, that you know. But at the same time, as we are learning in detail about all phylum, we must know this is a major characteristic which belong to all phyla. Therefore, it has to be understood. It cannot be neglected. So, zygote develop into 2 cell stage, 4 cell stage, 8 cell stage called morula stage. 8 into 16 cell stage, still it is called as morula. It is like a cricket solid ball. Each cell present in this morula is called blastoma. Why? Because that rearranged to form the blastula. The process of formation of blastula from morula is known as blastulation. See, this is the structure called blastula. Now, what is exactly we have to understand? These blastoma are arranged in such a way that outermost layer from trophoblast and inner from embryo mass. This embryo mass is the base to form all these types of pre-stages of embryo being diploblastic, triploblastic. Now see, these up to this stage, all organisms, they have common development of embryo. That means something is common among all, including porifera to human being. That means we are evolved from common. Therefore, we have some characteristic common in the early stages of development of embryo, that is known as ontogeny of recapitulation, given by Ernst Haeckel. Remember this word. This can be asked in NEET. Who has given theory of ontogeny of recapitulation? That is Ernst Haeckel. And what is it? Some of the stages in the embryo development are repeated by all the organisms. That is called ontogeny of phylogeny also, or recapitulation. I hope you got also called as biogenesis. Means some of the stages in the life cycle of even human being are repeated, that similar to porifera, cylindrata, platyhelminths, hemianthus, ascarimenthus, all other non-chordate and chordate. That is very important. So up to blastula, all organism has common development of embryo. But after blastula, how they get evolved? See, initially porifera and cylindrata, they are very simple, with only multicellular with tissue level organization. Porifera without tissue level organization, and cylindrate and ctenophore with tissue level organization having diploblastic acoelomate. What is diploblastic? See here. These embryo mass cells are arranged to form ectoderm and endoderm, not mesoderm. Means having only 2 layers in embryo, that is ectoderm and endoderm. Hence it is called diploblastic. Why it is called acoelomate? Because it has no true cavity. As you can see here, this is the cavity present inside the mesoderm of this organism. Presence of this cavity, as a cavity inside the mesoderm, they are called as coelomate. But is it there? As here it has mesoderm, it has no mesoderm. Since it has no mesoderm, there is no question of having cavity, therefore it is acoelomate. I hope you got it. So diploblastic acoelomate. So who are diploblastic acoelomate? So let us come to the first page. These 3, porifera, cylindrata, ctenophores, they are diploblastic acoelomate. We shall continue to understand what is triploblastic acoelomate. So triploblastic acoelomate. Let us understand first what is triploblastic acoelomate. See in this figure, this is triploblastic acoelomate. Triplo matlab 3. What are the 3? Ectoderm, mesoderm, endoderm. From which mass of cell these 3 layers are produced? From embryo mass. As I told you, this is the common feature for all. So from this embryo mass, these 3 layers are developed. As a evolutionary trend, here mesoderm was absent. Here mesoderm is there. Hence mesoderm was absent. Therefore here it is diploblastic. Here it is triploblastic. That major difference. I hope you got it. Presence of 3 germinal layer, ectoderm, mesoderm, endoderm, triploblastic. Now, mesoderm is there, but in mesoderm there is no gap, there is no cavity, there is no space to to have arrangement of organ system. So it is simply that layer is formed but no cavity in that mesoderm, therefore it is triploblastic acoelomate. I hope you got it. What is acoelomate? No doubt mesoderm is there, it has evolved in a evolutionary order, but still cavity is not developed, as here false cavity, that is called fragmented cavity, and true cavity is developed. Here there is no true cavity, therefore it is triploblastic acoelomate. Now who are triploblastic acoelomate? Come to the video. Here only one group of organism, that is Platyhelminthes, is triploblastic acoelomate. How it is easy to understand? See, these three, Porifera, Coelom and Ctenophore, Diploblastic acoelomate, Platyhelminthes, Triploblastic acoelomate. That is clear to you. After Platyhelminthes, now let us understand about Ascohelminthes, how they are separated from Platyhelminthes on the basis of organization of three germinal layers. Now for Ascohelminthes, let us observe this diagram. See here, this is the diagram where you can see there are three layers: ectoderm, mesoderm, endoderm, triploblastic. But see, in mesoderm, fragmented cavity is known as pseudocoelomate. Hence, this type of embryo development stage is called triploblastic pseudocoelomate. These Ascohelminthes in a order, they are triploblastic pseudocoelomate. Once again, Porifera, Coelom, Ctenophora, Diploblastic acoelomate, Triploblastic acoelomate, Triploblastic pseudocoelomate. I hope you got it. Now next, annelid onward. Up to human being, who are they? They are triploblastic coelomate. Let us understand. This is the highest development of embryo at that level, having cavity in mesoderm, that is called coelom, and that is the true coelom. Hence, these group of organisms, they are called triploblastic coelomate. Once again, ectoderm, mesoderm, endoderm. Hence, it is triploblastic. And see in mesoderm, this green color layers of the cell from out and inside, having cavity in middle. Here, this is a cavity in middle. This cavity you can see in the middle, that is called coelom, true coelom. Hence, they are triploblastic coelomate. So let us consider that who are diploblastic acoelomate. पुनः मैं बताना चाहता हूँ कि डिप्लोब्लास्टिक acoelomate कौन है? पोरीफेरा, सिलेंट्रेटा, टिनोफेरा. ट्रिप्लोब्लास्टिक coelomate कौन है? acoelomate कौन है? तो प्लैटीहेल्मिंथस. ट्रिप्लोब्लास्टिक pseudocoelomate कौन है? तो एस्कीहेल्मिंथस. एंड ट्रिप्लोब्लास्टिक coelomate कौन है? तो एनिलिड्स ऑनवर्ड सभी के सभी फाइलम में आने वाले ऑर्गेनिज्म ट्रिप्लोब्लास्टिक coelomate है. अभी इसका क्या महत्व है? Why it is to be considered in the evolutionary trend? इनके अंदर पोरीफेरा में टिश्यू लेवल ऑर्गेनाइजेशन नहीं है, बट सिलेंट्रेट एंड टिनोफेरा में टिश्यू लेवल ऑर्गेनाइजेशन आ गया. यहाँ पे ट्रिप्लोब्लास्टिक acoelomate में ऑर्गन लेवल ऑर्गेनाइजेशन आ गया, बिकॉज मेसोडर्म आने से ऑर्गन सिस्टम और ऑर्गन डेवलपमेंट की शुरुआत हो गई, लेकिन कंप्लीटली ऑर्गन सिस्टम डेवलप नहीं हुए. इसलिए मेसोडर्म के साथ ऑर्गन तो आ गए, इसलिए ऑर्गन लेवल ऑफ डेवलपमेंट. इन एस्केलेमेंथस फॉल्स कैविटी आ गई, फ्रैगमेंटेड कैविटी आ गई, इसलिए क्या हो गया, थोड़े थोड़े ऑर्गन सिस्टम बनाने के लिए जगह आ गई. इसलिए मेसोडर्म विद कैविटी दे स्टार्टेड टू प्रोड्यूस some incompletely developed organ system. But in annelids onward, because of complete and true cavity, that they have organ system level of organization. So once again, this type of embryo has diploblastic acoelomate to have the cellular level organization and tissue level organization, but no organ level organization. Platyhelminthes has organ level organization. Ascohelminthes has organ level, well developed organ level, but poorly developed organ system level. And annelids onward, well developed organ level and organ system level, because they have true cavity and they are triploblastic coelomate. So once again, come to the first page. As a part of classification, what we are understanding, these protozoa, they are already separated because they are unicellular. Metazoa, they are multicellular. Then under multicellular without tissue level, with tissue level. They are diploblastic acoelomate. Even coelenterates and ctenophores also diploblastic acoelomate, but they have tissue level organization, therefore they are separated from porifera. Now platyhelminthes are separated from ctenophora because they are triploblastic acoelomate with organ level of organization. Ascohelminthes, they are separated from platyhelminthes having triploblastic pseudocoelomate, therefore they have poorly developed organ system level of organization. And annelids onward up to human being with highest development, they are triploblastic coelomate, and hence they have organ system level of organization. And in that again evolutionary history that we are going to learn under the heading of that phylum. But I hope you would have got it, that how whole organisms are classified on the basis of that three germinal layer: endoderm, mesoderm, ectoderm, presence, absence, as well as presence of absence of cavity, acoelomate, coelomate. If it is fragmented, pseudocoelomate. I hope this basic classification, this basic characteristics, which is considered for the classification of whole organism, is very important to understand the whole organism, how they would have evolved. Because in the trend of evolutionary history. When we study how porifera evolved into cylindrate, how cylindrate evolved into ctenophore and platyhelminthes, how platyhelminthes evolved into ascahelminthes, ascahelminthes into annelids, and so on. For that it is very important. I hope you have got very important phenomenon that ontogeny of recapitulation. Up to this stage, all group of organisms have repeated stage, and as mesoderm and ectoderm and endoderm, that is found in all. So initially, up to few stages, some of the features are common in all organisms. That is the evidence shows that all organisms would have evolved from common cells or basic organism like protozoa and parazoa. That protozoa and parazoa was the beginning of evolution of life on the earth. I hope you would have known that around 3,500 million cells ago, when first cell could have begin to form, that develop into protocell, coacervate, prokaryote, eukaryote with cellular level organization, eukaryote with tissue level organization, then eukaryote with organ level, organ system level of organization, and so on. We are going to learn in detail about other characteristics also, how they would have been used for the classification of organism. So see in this chart. Another point which is used as a feature to classify this whole kingdom is symmetry. This is radial symmetry, this is bilateral symmetry. In radial symmetry, any organism body divided into two equal halves through more than one plane is called radial symmetry. Only two group of organisms, echinodermata and cylindrate, they are radially symmetrical, including ctenophore. So cylindrate, ctenophore and echinodermata, they are radially symmetrical. Rest of other are bilaterally symmetrical. As I hope you got it, what is the definition of radial symmetry? What is bilateral symmetry? When our body is divided into two equal halves through only one plane, not more than one plane, that is called bilateral symmetry. So we human beings, including the rest of the group of organisms except echinoderm, cylindrata and ctenophore, they are bilaterally symmetrical. What? Bilaterally symmetrical when body is divided into two equal halves through only one plane, that is bilaterally symmetrical. When body is divided into two equal halves through more than one plane, that is radial symmetry. I hope you got it, how to use this point. All organisms, you can use this point whether they are bilaterally symmetrical or radially symmetrical. So who are radially symmetrical? To understand, come to the page. Cylindrate, ctenophore and echinodermata, they are radially symmetrical. Rest of other are bilaterally symmetrical. How it is easy to use this character to get the half mark? Because you ask question, write any 6 characteristics of phylum, 4 characteristics of phylum. In that you can use diploblastic, coelomate, acoelomate, diploblastic, acoelomate, pseudocoelomate, or bilaterally symmetrical and radially symmetrical. So that this will help you to get the mark. Also you understand that second characteristic, symmetry. Now third characteristic, basic characteristic which is used to classify the organism is segmentation. Metameric segmentation, non-metameric segmentation. What is metameric segmentation? Segmentation of body, that means part of the body after equal of interval of distance is called as metameric segmentation. If that segmentation is unequal, that is not equal after not regular interval of time, that is called non-metameric segmentation. Here all annelids, they show metameric segmentation, insect they show non-metameric segmentation. I hope you got this point. So arthropods are non-metamerically segmented, annelids are metamerically segmented. Another point for classification. Open system, closed circulatory system. All non-chordate, they have open circulatory system. All chordate, they have closed circulatory system. What does it mean? It means that blood come out in the cavity called hemocoel, because there is no continuity of capillaries and venules. Therefore, blood come out, and from that blood, oxygen is distributed by cell and carbon dioxide added into blood. Further, that blood enter into these capillaries, venules, and then come to the heart through vein. That heart is called venous heart then. But here you can understand there is a closed circulatory system. When blood flows through the artery, it is in continuation with venules, and therefore blood do not come outside. But oxygen diffuse out and carbon dioxide diffuse in, but blood is not directly in touch of cells. They receive oxygen and deliver carbon dioxide by diffusion. This type of circulatory system is called closed circulatory system. So you can use this point for non-chordate. All they have open circulatory system. For chordate, all they have closed circulatory system. I hope you got it. Very important point. Next characteristic feature, blind side body plan, tubing to body plan. If they have only one mouth or only one opening for ingestion and ejection, खाने के लिए और एक्सक्रीटा निकालने के लिए एक ही ओपनिंग होता है, उसको बोलते हैं ब्लाइंड साइड बॉडी प्लान एंड ट्यूबिंग टू बॉडी प्लान. Those organisms who has mouth to ingest the food and anus to excrete the food, that is called tubing to body plan. So these also characteristics you can use for ascahelminthes onward, because ascahelminthes onward they have tubing to body plan, but porifera, cylindrata, ctenophore, they are blind side water plants. See how easy it is to use as a character when we have to write about character of the phylum. So here cellular organization, in that cellular organization, two major things again you can use. One, open canal system or water canal system. Only porifera has water canal system and Echinodermata has water vascular system, and that we are going to learn in detail when we learn about that phylum. I hope you got these some of the basic characteristics which supposed to be used to classify whole animal kingdom. And with this chart and that explanation, you please find out that photo with this video when we will have this presentation of this video, or we will put this video on our channel. I hope you would have watched it, you would have enjoyed it. You please continue with this video as and when again and again if you see, you will definitely get it whatever I explained with the diagrams given with this video. I request you and appeal you to like and share our channel, as well as if you are new then subscribe it